So I have a list of students' name here. So today is our first lecture. So I want to give a brief introduction, what will happen for this semester, and what we will do together, and what will be the requirements for the courses. And we try to put everything on the course website, SEBA. So if you already familiar with the system, I think you know how to get all the information. But I'll emphasize some of the important points in class. I think maybe some of the students probably know when we do long distance course, usually it's not to start from 10 past 9. But I just checked on my course website. I didn't specify that. So that means you have to be here on time, like 10 past 9 for today. So that's the expected. I want you to know what I expect to like a student to participate. But starting from next week, the courses will start from 9:30. I will explain to you about that. And every lecture will be recorded. So then we will go back. So if you want to go back to some of the materials, and you can go to the YouTube channel and to look into to refresh your memory. So okay, so I'll like to start it. So we have by this morning we should have 15 students enrolled in this class, but right now we don't have that many, so we'll see what's going on. But this is course, you can see, as I mentioned here, this is what we call frontier sciences in biotechnology. And if you see my syllabus, you know this is a long distance course with Kyoto University. And I'm the coordinate, so I will do a few lectures and try to work with you together on some of the projects. Then I will participate in most of the lectures. Then we'll do something together. Okay, so this is about the course. And the course actually started in Kyoto University a few years ago. The course coordinate is Professor Wang. And Professor Wang is a young scientist in Kyoto University. She appointed by Institute for Integrated Cell Material Science. And she's the one initially started the course for graduate student, both master and PhD student. And according to what she mentioned to me is most of so when she started the course, actually there's also very few students, but now this become a very popular course at Kyoto University. So what we usually do in a class is we will invite faculty members on either NTU campus or Kyoto campus. Then we'll ask them to deliver a lecture. Then after their lecture, we'll have questions to discuss. So then we'll tell you how we are going to do that. So she initially set up the course and become very successful. Then I will give you a few examples what they did in the past. But this semester is the first time we're try to do this long distance course among like 3-4 long distance course under the Center for Biotechnology. Then, as you can see here, the time for the course in Japan, starting from 10:30, which is 9:30 in Taipei. So usually we do, let's see. Usually we do the first lecture according to NTU campus time because Kyoto University's semester won't start until early April. So this is how, so that's why I say I explain I usually start on time. So I know I'm 1 minute late today, but usually I start on time punctuated. And it hasn't happened. There's no student show up. So I was able to do that through my career so far. So it's on Wednesday here. Like obviously you probably all know this. Then we will get together, and there's a video recording in this room. Not only the screen and myself, so you can see in the middle. And this is the screen in the middle. This is the screen what Kyoto University students and faculty will see. So for like slides and the speaker. Then our screen will be on both sides. So you can see the screen. And if it's not clear, we'll try to ask our faculty member to upload their file as soon as they are ready. So hopefully you also can download the file from the course website. As I told you, there are many cameras in the room. So what you did in the class was like a completely recorded. We cannot control that. So the cameras just surround, and this is a part of that. So when we have student presentation or question sections, and please use your microphone. So there are microphones on the desk. So when you want to speak, ask questions, or we'll have group discussions, we'll ask you to present some of the conclusions. And so in order for our partners to hear your voice clearly, so please use the microphone. So in the room, you say, "Oh, I can shout," so everybody can hear me. But you have to think about student and faculty on the other like a campus. They need to like hear you clearly. So try to make like your song clear enough in order for all students to participate. And in our campus, as I say in the beginning, we probably have like 15-ish students participate in this course. At the Kyoto side, based on what happened previously, it's probably will have 60 to 80 students enroll. And most of them are graduate students, just based on what happened last year. So starting from next week, we'll meet at 9:30 here. So the room will open like I don't know 10 past 9. Actually, it's more like around 9 o'clock. It's already ready for the course. But just to make sure we know the time to meet every week, so I'll do 9:30 Taipei time or in like a national standard time here. So 9:30 to all the way to 11. That's the time period we will be connected with Kyoto University starting from April. Then, well, of course, we have some buffering time to discuss if you have some issues. And also we can utilize the time between 11 to 12 o'clock for group discussion. So then we'll see how that goes. So this is like a basic setup for the course. And some of you probably already seen the syllabus. So this is what I want to show you. We did this together. Actually, we start discussing this class when I think in December, last December. Then we quickly meet in January to set up like the possible arrangement between two campuses. So in March, what we'll do is I will describe what I expect to do for the first 3 weeks. And in these 3 weeks, we will review some of the what I call like a landmark papers or experiments in molecular biology. And this is sort of like the foundation for everybody understand what we are going to sort of like look forward. So then I also need your participation. Then in mid-April, this is the time we'll start doing the long distance course. So April 18 is the first day we are starting the long distance course. We'll have we invite faculty members in National Taiwan University or Kyoto University to deliver the lecture. So the lecture itself will base on faculty's expertise and to try to bring up like the new concepts or approaches or sort of like setups in this field. So every week will be slightly different topics. So they already provide their talk. Then we will ask them to provide some readings if necessary. So this will be, so then I use different color to code like which side will be the main, sort of like the instructor. So then if we have faculty from NTU, then you will see professors here to talk to you. And if the lecture is going to be delivered by like faculty at the Kyoto University, then we'll see the middle screen. So like this one. So today we have both, but it's all in Taipei. So in like starting from April, the middle part will be, for example, the Kyoto University's professor. They will deliver a lecture. So you'll see the professor and part of their classroom, part of their screen in the middle. Then I think in the middle, we also can control. Sometimes we'll see the audience, so the students in the room or the student in our room. So both can be arranged. And on the side will be the screen, just like this one. So today we're just like mimic what will happen like when we start a long distance course. Okay. So this semester is very long. So we can go all the way to June 20th. But actually, if you know the semester in Japan, actually the semester won't end until sort of like July. But for this semester, we ended this like the last week. The last week of June is the final week. So the last lecture I listed here is June 20. But if everybody would like to participate, then actually June 27, there's a lecture too, and also in July. So we'll see how we can arrange that. But officially, the class will end June 20th as we scheduled in NTU calendar. Then the very last week, like the last date of June. So this is a tradition with the long distance course in Center for Biotechnology. So we collaborate with Kyoto University. We host mini symposiums, so for mainly for students. Different years we did differently. So in some years, Kyoto University students and faculty will visit Taipei. But this year, we are going to Kyoto University, but not for the whole class, because we don't have the budget to sort of have the whole class to Kyoto University. And also, we have 3 to 4 long distance course, so we have to have a selection system like a procedure. But we encourage students, especially like graduate student, you already have some research results, because for the mini symposium, we assume student will present their poster or a talk at the mini symposium. And the purpose for the mini symposium is for students to communicate with each other between the two campuses. So we usually do that. And this has been done for the past 10 years. Then in different semesters, so this is spring semester. In the fall semester, we will have Tsukuba University of Tsukuba join us. So in the fall semester, actually 3 campuses join the long distance course together. And we have many symposiums. Sometimes we also invite students and faculty from University of Tsukuba. So this is what will happen. Then we already know the date, so I put the date there. So June 30. Then we will have 2 to 3 faculty with a student sort of like attend this mini symposium. And more information will be posted with our course, and also campus-wide will announce this opportunity, because for students, they participate in a long distance course in the fall. They might not be in the class this semester, but we would like to inform them, because this is sort of like a campus-wide activity. So all students participate in the long distance course can apply to attend the mini symposium. So this year, the date already set up is June 30. So you have to mark your calendar if you are interested. And of course, we'll ask you to have a certain level or English proficiency. And also we like to, because it's a presentation, so you need to submit a research abstract. So the abstract has based on your research work that here. And probably we also need your advisor approve. He or she is willing to allow you to attend the mini symposium and present your work. So there are some requirements for that. But if you are a graduate student, especially second year master's student or senior PhD student, we encourage you to think about, put it in your plan, like on your calendar, then think about you might want to participate in this activity. Okay, any questions so far? Okay, so then let me go on. So this is what I put in the course website, what we will do and what we expect to do. So you can see here, its important thing is we will set up some fundamental, we'll introduce some fundamental concepts. And because we try to invite all the young faculty members on campus, on both campuses, then we expect some of them will bring new technologies or new concepts or new experimental approaches in our field. So this is where we try to set up the course and try to allow students interact with our faculty members. Then not only we ask the faculty deliver the lecture, we also want students actively participate. So this is what we did before. When I say we, means the course did before. Okay, so we'll have small group discussion and some activity. So what we did before is at the end of every lecture, we have discussion questions posed to the audience. Then later today, we'll divide students into different groups. So basically 4 to 5 students will form a group. And we try to separate students at a different level. So we want in each group at our campus, we want to have undergraduate students, have master student and PhD student. We don't want one group with all PhD student and the other group with all other like undergraduate. So we try to separate students with different background. So for example, we have students from Department of Mathematics and from Department of Life Science, Department of Biological Chemistry and Sciences. All those we want to divide into different groups. Then we'll do that after we make sure all the enrollment list. So this is then for the small group discussion. If we are only one classroom, so what do is we ask the group sit together. Then you really discuss with your partner on that specific question. So we'll give you a certain period of time, for example, like 10 to 20 minutes. Then we'll ask you also, you have to have a leader. So you have to have by the end of your discussion, in your group, you have to have one leader and to stand up or just sit, then use the microphone to tell everybody, including students in the Kyoto University, what's your conclusion about that specific question. So we'll do that on both campuses. So at the end of the discussion, for example, here we might divide everybody to 3 groups. Then each group will speak 1 to 2 minutes. So you have to be really concise. And this is how we are going to do that. And in Kyoto University, we might have, I don't know, the number from 8, 10, or 12 different small groups, and they will do the same thing. We will hear their conclusions through the screen. So this is how we are going to do a discussion and just like a small group discussion and some activities. So then because of that, you have to work as a team member. So you work with your partner and try to like focus your discussion as the like and also organize your discussion, because you have very short time to talk to all the audience. Okay, so this is what we will do. So every week we have a one lecture and a group discussion, and at the end you will hear all the participants, what the conclusions from all the participate students. Okay, so this is, and I know this might be new for some of you, but it's new for me too. So we'll try to do that, and it's new for this class because we are going to do like on both campuses. So we'll see how that goes. But this is what we decide. Okay. So then what should we do in March? So I don't like to have a blank month. So I think you pay for the tuition, and I have to do something with you together. So what I decide to do is I'm not going to lecturing for entirely 3 weeks, but I would like you to participate in some of the work. So if you see my syllabus, I say, okay, let's look at some landmark experiments or landmark papers together, and let's work out something. And in order to be more efficient, I also want you to work as a group. But I want to show you, before we go on, I want to show you what the previous student did for this class as a homework. Okay. So at the end of the semester, we used to ask students to write a term paper, very short term paper, a few pages. Like I look at the previous students' term paper is about 2 to 3 pages. Most of them probably is only 2 pages. And I put the course website here. You can link it to the course website. You'll see that. So this is the course, like where can you find the previous homework? It's like under the Professor Wang's homework, like a website. So this is her website, and if you're interested, you can look into. But there's a homework assignment for this course. So under the homework assignments, you will see 2 links. And these 2 links, one is 2016, the other is 2017. And what she did is she already compiled all the files into all the students' term paper into one big file. So once you click on it, then you will see a paper like this. It's not exactly like this because I want to show you on the PowerPoint screen, so I sort of modify a little bit. But every word was written on the term paper. So students were asked to write a short essay on my favorite experiments in science. So it's not only in biology, but in science. So it's a sort of like give students a broad range of things they can explore. Then, of course, every student will write on one specific topic. So the one I put it here is this famous Mendelssohn and Stolz experiments on DNA replication. So a lot of you probably learned it in high school, and also we refresh our memory in college as well. So this is quite important work in the history of life science. So you can see they have a short title. As we write the term paper, we give a title, right? Then he or she describe what's about this experiment. And of course, this is just the beginning of this term paper. Then most students will have figures go with the paper. So the figures usually illustrate the original ideas or hypotheses. And in this particular case, on the second page, the student also present the expected results just by drawing the possible, like 3 different possible cases. So those are the student did previously. So I just want to show you what the student did previously in the class, and at the end, they focus on one experiment and present this, like present it to turn into the class as this one. So of course, this is very nice, and you put in a file. But I doubt how many students will go through the entire file from page 1 to the end. If you think about, if we have like 30, 40 students, then each student writes 2 to 3 pages. And it's a huge chunk of reading, right? And it's easy for you to sort of like a flip through. But I was thinking about maybe there's a more efficient way to present this type of work. So what I had in mind, I want to show you is some of those websites or papers I like. So the first thing, if I put this like landmark experiments in molecular biology, if I put this, and there's a book so under this title. And in the book is also every chapter, they describe one experiment in detail, give you some background information, what happened or what was known at that time, and what was the questions scientists would like to address, and how they set up experiments and to solve the question, and what was the end results. So this is sort of like a typically people, especially for young students, you want to get into a field. I think it's very good reading and very informative. So that's one type. And the other is something like this one. So this is a website set up by Cold Spring Harbor Laboratories, and it's DNA from beginning, and it's all centered around genetics concepts. So I teach genetics starting from my first year of my career. So I started teaching genetics and biology for a very long time. So you can see this website, they separate different experiment into 3 huge classes. And if you took genetics before, and you are probably familiar with those terms. So they put experiments under classic genetics, under molecular genetics, and under some other like items. Then I think the important thing is they organize the information as what we expect, and you can use that, really learn genetics by yourself. And this website has been set up for a very long time. So I just took one example here. So this is like I click on one particular experiment. So they have the specific concept. So they organize the experiment into the major conclusion, what they call concepts. And this concept is about one gene, one protein. So then they mark their concept clearly as the title. And of course, inside there's like a short article explain this concept. Then you can also, there are many choices. You can look into their animations about how the experiment was set up and was conducted, and what's the major conclusion. And on the side, there's a sort of like a list of concepts under this big item. So this is under molecular genetics, and they list several concepts. For each concept, you can zoom in to see what was being done, what has been done previously, allow us to understand this specific concept in biology. So, and I usually give the website link to my general biology student, the freshman students, for them to explore some of the genetic concept, because usually I teach genetics for undergraduates. The other one is several years ago, I started teach developmental biology laboratories. So I'm only responsible for 1 or 2 weeks of laboratory courses. Then I use the nature milestone webpage. And under the milestone webpage, there are several different specific fields. So there are subfields under this, and development is one of them. So I want to show you what's in there. So this is what I usually show students when I introduce sort of like developmental biology to students. And this is how the field starts with experimental embryology in the beginning. So then they are like the field progress with different landmark experiments. So I separate the timeline into 2 columns. So you should start from your left-hand side here, all the way, then you start from here, you go all the way down. So this is how they pick up several different experiments in the past, and they give a specific time. This is how we usually do. So we usually like to organize our knowledge based on the development, so the history of the development. So this is how, so they have like several of them, then indicate which year, who did what experiments. And if you click in, then you will see something like that. So this is like the second one. So it's to do lineage tracing. So then it's like if you learn development or cell biology, I think you are familiar with the term called cell fate. During embryo development, and actually cell differentiate and become, so we all like animals or plants like us, we all start from a single cell to multicellular organism. And during this process, each cell has to be specified for its role in the final sort of like an organism in the whole body plan. So this is, then you can see the milestone. They organize 24 of them. Of course, you can do more, but they pick up 24 important milestones in this field in development. Then they organize by like the time. So see the milestone 2, and this milestone happened in 1929. So that's the year. Then also, as you can see, every different organization or student did is the same. We give a short title, right? So this is the key things we're going to sort of like describe here. Then very similar to my favorite experiment example, I have like short description and some figure or illustrations on the side. Most importantly to me, I think most importantly is if you read through the article usually is not very long. It's like a mini review. So probably only 2 to 3 print pages. So it's not a very long article. But at the end of the article, I like they organize original research papers and also some further readings. Okay, so the research papers are, because this concept was set up or was developed during certain period of time. So see, the first paper here is the classic paper published in 1929. And this is how exactly this technique was set up back then. And they give the link of this landmark paper. So you can see this is the first paper listed here. The same technique has been improved through the years. So several decades after the first publication, then they list another one here for you to look into it. So both of them are important sort of like a landmark experiments in the field. So they put that as an original paper. I think in biology it's very important to know how those concepts was developed in the history, and also what type of experiment was done, or what are the evidences support this idea. And this is what we do. And usually the early papers, the first papers was really important in the field. And it could be one specific experiment. It also could be one big improvement of instrument. So technology wise, also lead the field into a different era. So this is how, so I think students, if you are interested in the field, you should like really look into the original article. Sometimes the original paper is not very easy to read, but it's a challenge for you, especially for graduate students. If you are really interested in exploring the field, then I think it's important for you to know what was done before. Okay. Then the second section at the end, so this is toward to the end of the article that list further readings. So the readings sometimes are like review papers or long or short or some other related knowledges for this one. So those are also if you want to know more and there's more readings you can do. But I will encourage some of you really, no matter which concept or which experiment you really enjoy and you think is fascinating in the field, and you should really dig into their original paper. So, of course, you cannot do every concept like this one, but for the one related to your work, like for graduate student, you are working on a specific field, and I think you should be familiar with the background information. And those original research papers are really, really important. Okay. And this is another example. And this example is about Human Genome Project. And I think many of you probably seen this poster. And this poster actually tell you the development of genetics, start from Mendel. Then this is how Human Genome Project started. And actually, compared to the long history of genetics, our genomics is only at their infancy. So you can see here, like within this short period of time, so this poster was done while we finished the human genome draft. So start from 1990 all the way to 2003. Of course, there's a lot more technology development, knowledge synthesis after that. But this is another example to show you how people can organize the development of a field and into sort of like a good presentation. So this is what I would like to encourage you to do for this semester. Then you probably will be scared. Are we going to do like the entire history for hundreds of years? We are not thinking about that. So I want you to, as I say, we'll separate into small groups. And I want you to do is within your group or among all of us. So we have 15 people here. Then we'll think about doing something like this one. Then what we would do together is to put one specific thing for the entire course. It could be molecular biology or genetics or whatever you prefer to do because it's like a group project. I'm the one just like I'm participating here. You can consult me if I know the field more. If I don't know, I have many colleagues are very good in different subfields in biology, so we can always get help. But no matter what we would like to do, we'd like to sort of conduct the project as a big group. Then I'm expecting to have a project like this one. So we'll have a timeline. It doesn't have to be have to be as complicated as this one. It could be very simple as the landmark we see in development, like this one. So we'll have a timeline starting from, I don't know, 18th century or 19th century. It depends on the field, right? Then from then to now, 2018, based on the thing we would like to do, and we have landmark papers listed by a short term, like this one. So for example here, every landmark paper, for example, the second one is fate mapping. So cell fate. How do we know cell has different fate? And how do we know a group of cell, they inherit the same cell fate, and how the cell fate was determined, right? First, we have to observe whether there's a cell fate, and then we can set up something to understand how cell fate was established during development. Okay, so this can be just the time, the year, and the concept or the technique. Either way. So then we have to do it together. Then we have a timeline from the beginning to the end. Then we'll have many like short phrases here. Once we have this, then we'll have to do the second thing. The second thing will be done in small group. So if we have 4 or 5 students in one group, then given the timeline in this way, they will say, okay, we would like to do something, one particular experiment in here. So for example, here, we can look into it. Then I say, oh, maybe I'm interested in this one, asymmetric cell division. Oh, this is very important. And we have faculty member will talk with you her research about stem cell and how we can apply that to future medicine. So say, oh, this is really fascinating. So I want to do this. And since we already have a specific term or concept or experiment listed there, then I can zoom in to read the original paper, to read textbook about this concept, to read a lot more information, especially for really famous experiment. You will find many information online. And the challenge for you is how you want to present to all of us. Then you have to think about a way to organize it. So we want to have some text content. So you have a short essay about this one. And we can have some illustration from the original paper, or you redraw it. Then we also, if you want, we can have some short video. And there are many videos introduce different concepts in biology. And you can find the good one, make a link, let us know what are the good video or lectures about this concept. Or you would like to deliver a short one for 2 to 5 minutes. So you have a lot of freedom based on that to do. But you say, I don't want to do, I'm not very good at like a computer or something. So maybe we are on the same, sort of like at the same level, at least we can complete a short essay as what we previously do. So basically your like basic requirement is focus on one essay and finish that, just as we can check what previously did. So all this so we first need to decide a thing for the whole group, then for the whole class, so for 15 people, then we'll do small group. So we'll do this together. We can split the effort how you would like to do it. Then we'll have a specific concept. So we depend on how many students we have. We might not be able to complete every concept. For example, here we have 24. If each one of finish one, then we'll only finish 15 out of 24, which is okay because this will have a course website, and under the course website, we can always trace back. So for example, if the next semester, not next semester, next year, when I like restart the course like today, then I will be able to show the new students what was done this semester. Then maybe some of them like your idea and like some of the experiment listed on the screen, then say, okay, we decide I want to do this one. So they can add it on this timeline. Then we'll in one will like a complete. So this will be, I think this is not only allow students to do one landmark experiment in detail, in depth, but also organize in a way for other people can learn this field. So your contribution will be there for like others. I think that's something I would like to sort of like lead or work with you together and to accomplish. Okay. Any questions about this? Because this is by the end of the semester, this is what you need to finish. Any question about this? Okay. So if there's no question about this, then I assume all of you all understand what happened. For a student, you come in late, I just want to let you know everything will be recorded. So if you focus on your cell phone, unless you're watching my slides, will be entirely videotaped. Then if we have professor, if I'm not the instructor, I will sit in the back. Then if I think you're not really focusing on the class, I will give you a small sticker to remind you. So you don't want to get a sticker from me. You know, in elementary school, if you behave good, then professor, like the teacher will give you a sticker as a bonus, as a sort of like a reward, right? We're not doing that. We're not going to give you a sticker. I'll give you a piece of paper, remind you. I don't think you are focusing on the lecture. Okay, so this is why. So I would, if I'm here, and also you have a TA sitting in the back, then we are watching you, and the video are recording all the time. Just to let you know, and you know, if one of the camera really zoom in and show up, and you know, your behavior could go over the Kyoto campus as well. Okay, so this is just, and try to be on time. Okay, so any question about the course? Okay, so I spent an hour to do this, so I'm going to do, so I'll take 10 minutes break. So usually if I do lecture, I do like on time. I usually watch, say, I have my computer here, usually is for looking at the time because sometimes the clock is not like right in front of me, or sometimes when I speak to like get into my lecture, I forgot the time, but I try to be. So I'll take 10 minutes break and come back. I'll let students introduce yourself to the class. So we have to know each other. Then we'll divide you to 3 groups. As I say, you know, we will divide graduate student, like a PhD student, into different group, and undergraduate into different group. So we'll do that for the second hour. Then I will allow you know each other within your group and discuss what would you like to do. So today we'll finish a lecture, finish today's activity at 11. So then we'll do that so every sort of every week. Any questions? Okay, let's take 10 minutes break.